Hello. My name is Alejandra Bochabat. I'm at Icahn School of Medicine at Mount Sinai in New York. And today I'm going to present a work done with Dr. Medicine Potash at Polsky Lab in collaboration. And it's about how innate immune responses reverse HIV cognitive disease in mice and profiled by RNA sequencing in the brain. Then first, I would like to talk a little bit about HIV-related cognitive impairment, or NCI, and innate immune responses. And as all of you know, despite antiretroviral therapy, half of infected people develop cognitive impairment that persists for life and worsens with age. The continued expression of viral proteins like that or GP120 in chronically infected cells in the brain is considered to be one driver of neuropathogenesis, and it's for that we need interventions that target HIV expression. Also, host antiviral response like innate immunity control can eliminate many viral infections, and one of the best known innate immunity responses are toll-like receptors that recognize ligands derived from pathogens and signal for changes in gene expression. Then in this work, we use the compound poly-IC, which is the toll-like receptor 3 ligand to induce immune response. And we want to see the effect of poly-IC in HIV NCI, in virus load, behavioral defect, and also in transcriptional changes in the brain. And for that, we are going to use we use the eco-HIV infection in mice. And I guess many of you know about this virus, the eco-HIV. Then this virus, it's the protein, the envelope protein from the HIV human virus, GP120, is replaced by the GP80, which is the envelope of the murine leukemia virus, or MLV. Then this chimeric virus is able to infect regular immunocompetent mice. As we see here, viral DNA copies in spleen cells 1 month and 6 months after infection, and in less extent also in brain tissue. And one of the characteristics of this virus is that it produces a behavioral defect in the infected mice, usually around 1 month after infection. And you can actually test or assess this behavioral defect by different behavioral tests. And one of the ones that we most used in our lab is called the radial arm water maze test that you can see here like an example. It's maze that it's covered with opaque water, and we put a hidden platform in one of the arms, and the mice have to find it. And here, actually, I show an example of the results. Then we do that during multiple days, and we average the last 3 days, and we count the number of errors for the mice to find the hidden platform, and we perform different trials during the day, 4 trials and a retention trial that is half an hour after the last trial. And here you can see that the eco-HIV infected mice perform significantly more errors when compared to the control mice. And that is 1 month after infection, but we see the same defect 6 months after infection. As you see here, I just plot the last trial and the retention trial here, but we see that the infected mice perform more errors than the control mice. Then what did we do? We actually perform infection or infect the mice by intraperitoneal way with eco-HIV and PBS. And we wait for a month which is the time that, as I told you, it's more or less needed to develop the cognitive impairment in these mice. And then we start the administration of poly-IC, also by IP injection, and we administer 50 micrograms 3 times a week. Then we wait for 2 weeks. The last week is the behavioral test. And then we isolate the brain and specifically dissect the striatum to perform RNA sequencing. And first, I'm going to show you the results for the behavioral test. And here, as you see, the echo HIV, which is the red one, perform different or like higher number of errors in finding the hidden platform. And when we look at echo HIV plus this compound poly-IC, that is the green one, we see that they actually perform almost as the controls. And this is even more clear when we look at latency. As you see here, there's very different, the infected versus the treated. And the latency is actually the time in seconds to find that they need to find the hidden platform. Then we can say that poly-IC administration restores cognitive function in infected mice. Then we also look at the virus burden in eco-HIV infected mice with and without poly-IC treatment. And here I show the RNA-seq RNA copies per microgram RNA for eco-HIV and eco-HIV plus poly-IC in macrophages and spleen cells. And you can see that poly-IC administration blocked virus transcription in these 2 cells. We also look at DNA, DNA copies per million cells, also HIV poly-IC into these 2 cells, and we see that poly-IC blocks DNA synthesis in macrophages, and we have a reduction in spleen, which is not significant. Then we perform also bulk RNA sequencing in striatum, and this slide is just to show you the number of dysregulated genes. And I don't know if you see here, but we choose a regular p-value of 0.05 and a fold change like higher or lower than 0 for up and down regulated genes. And when we look at poly-IC versus controls, we have like around 500 genes dysregulated. And the number of dysregulated genes is higher when we have the virus alone versus the control. And when we have infected mice with treated with poly-IC, the number of dysregulated genes goes down. And this only shows that the 2 infected groups with treatment or without treatment, we have a lot of the significant dysregulated genes. And actually here I show more or less the same thing, but I think it's more clear. Then we have like a big dysregulation in the striatum of like infected mice with eco-HIV, and the majority of these genes are downregulated. And when we administer like treatment, the number of genes is less or goes down. And that is just an example of a heat map representing the expression for each mouse. Then for significant genes in eco-HIV, and we can see that when we add the poly-IC for these genes, the expression goes back very similarly to the controls. Then these results suggest that poly-IC administration normalized gene expression in eco-HIV infected mice. And we also look at gene ontology analysis, and I'm not sure if you can read all that, but I mean, I can guide you through it. Here we have eco-HIV versus PBS, dysregulated pathways, eco-HIV plus poly-IC, and up and down pathways. Then what I want you to kind of see is that we have a big activation of interferon-related response in eco-HIV plus poly-IC. Like we can see here a lot of this interferon and other innate immune response activation. And when we look at the eco-HIV alone versus controls and downregulated, we see a very significant downregulation in neuronal function. I'm not sure if you can read the pathways, but almost all of them are implicated in neuronal function, like a neuronal system interaction at synapse, neurotransmitter release, and others. And the important thing is when we look at the downregulated pathways in eco-HIV that receive treatment, we don't have any of this downregulation in neuronal function. Then we think that poly-IC treatment reduces the eco-HIV dysregulation in synaptic function. And that is just an example of like this synaptic dysregulation that I was telling you that we see in eco-HIV. And then when we have the treatment, like we have a restoration, partially but significant, of many of these family genes, like neurotransmitter release, and also synaptic GABAergic or glutamatergic response or axon guidance.
3 minutes.
Okay, then also, as I told you, we have like a big interferon response, like we see in both treatment groups, especially in eco-HIV poly-IC, and we can tell that poly-IC induces a robust innate antiviral response in the S3 arm of these mice. We validate by qPCR, like many of the interferon genes and synaptic and other immune-related response genes in the brain. And then as a conclusion, we can tell that eco-HIV infection of mice cause cognitive impairment and dysregulated brain transcriptome, primarily at the level of neuronal function. However, we show also that the dysregulation is reversible. And poly-IC induces a robust innate antiviral immune response in the brain of chronically infected mice that reduce systemic virus burden. We see also that reverse cognitive disease and reduce the eco-HIV transcriptional dysregulation in the estriatum, specifically downregulation of synaptic-related pathways. And the future direction, we would like to evaluate requirements of selected genes for poly-IC antiviral action by knockout or other pharmaceutical interventions. And we would like to evaluate the poly-IC treatment in the context of antiretroviral treatment, and also in the presence of opiates in eco-HIV chronically infected mice, specifically with a focus on gene expression, and also trying to understand the poly-IC mechanism by maybe chromatin remodeling techniques. And I would like to thank all the lab, also David Volsky, but especially Dr. Potash, who started this project, and Bao-Chu Dong, who performed all the virology and behavioral experiments. Thank you.
Yeah, thank you. So we have time for a question or two, and we have one in the chat. So the first question is RNA-seq straight and fold change. So in your volcano plot cutoff, they were selected as 0. So most other studies select more than 1. Could you explain why you picked that cutoff of 0?
Yeah. Then we did. Actually, we wanted to. We performed this analysis. We also look at the ones, and we use to, for example, when we are more interested in one gene, we want to look at what is exactly the fold change, but we did all these different, like greater than 0 or than 1. But because we didn't have a lot of dysregulated genes, like I should choose that one to show, but it's not the final. Yeah, yeah. sorry. I mean, I don't know what's going on with my, okay.